Right. So essentially, we have also a channel on Teams that is called Workshop Overarching Questions. So please do use this channel to type up your contributions if we don't have them covered already. So what we have done during the workshop is we have tried to highlight what Tom and I have selected as some point of answers for these different questions, and you can see that we have already a few points. And what I would like to do collectively now is to go for 7-8 minutes on each of the points, and try to add to the list or to see if we can have an open discussion about each of these questions. And I would like to start with the first question, which is, what do we need to make DNA, RNA, nanotech in living cells more predictable and usable? Yes, I can see this, Tom. So Tom will be taking notes as we go through these questions. And I invite you to basically have a look at what we've already captured. So in the first day, we heard about William Huck's automatic experimental design parameterization toolbox, and will this help in basically designing and building DNA and RNA nanotech in living cells? Is there a limit to how far you can get with such a setup? Is it always going to struggle against natural stochasticity, or can it be quantitative? Should we be looking to use chimeric systems, cyborgs, half living, half synthetic? A little bit along the lines of what Oscar Sess told us during his talk. Will this make them more predictable? Should we use simplified environments for prototyping and characterization of DNA, RNA, nanotech in cell-like environments where we strip away some part of the complexity of the cellular environment, a la Richard Murray? Should we use machine learning or first principle modeling, or a combination of both? Can we use computational tools for predictive design, verification, and realization of nucleic acid circuits in living cells? Will crowding effects, which we have heard about in a few talks, allow us to make more use of spatial structure, having spatially resolved reaction diffusion processes? And finally, a provocative question that we also extracted from this meeting is, do we know enough non-equilibrium physics? Do we know enough systems chemistry? Is it just an engineering problem now? So yeah, this is what we have captured. Please tell us what you think, and how do you feel this workshop has contributed from your point of view to answering this very first question. And I leave the floor open to anyone that wants to jump in at this point. So please, if you want to tell us.
Yeah, someone's raised a hand, but
I can't see that on my screen, unfortunately.
That's okay. Okay, so somebody's raised a hand, so just let's let them speak. So it's Andrew. So Andrew, go ahead.
Well, from a rather ignorant point of view, I don't think it's a question of knowing more physics or more chemistry. I mean, the environment is horrendously complicated. So the one thing I've taken out of this workshop, which is actually positive, was Domitilla's talk about feedback. I mean, I think that the chances of putting a circuit in a cell that works in vitro and expecting it to work in vivo or in the cell are basically zero. But if it's engineered to be robust by embedding it in a feedback network to correct against the various perturbations that you can anticipate, I think you've got at least some chance.
Yeah, I think I would agree with that, especially because of my bias being a control engineer. Yeah. Anyone else wants to emphasize a point here?
So biological systems have. Hello. Can you hear me?
Yes, we can hear you.
I was going to say with feedback, definitely important, and it's definitely biological cells use it. My concern is that you end up, and I was just having a chat with Mustafa actually, and he was saying, you know, we've implemented these feedback circuits, but you have to do a lot of fine tuning to get the feedback controller to, which is not surprising. And I do worry to some extent how much you just push the problem back a layer, if you see what I mean. How do I make my feedback controller work?
Yes, I think that's true. But you could argue that if you have some form of understanding of the type of perturbation that act on your system, once you have tuned your feedback system, it should buy you robustness in different contexts and against different classes of perturbation that you might have taken into account via the feedback design.
Right, but traditionally, when you do feedback control, you would assume that the parameters of your controller are you're not needing to control the parameters of your controller. So
you would still need to tune it.
Anyway, there's lots. Lee and Eric have got their hands up, so let's let other people weigh in.
So Lee or Eric?
Eric?
Okay, sure, me. So on this robustness issue that Andrew brought up, I think one of the things that has really surprised me about the field is that there are certain kinds of molecular systems that we sometimes have designed, and I'm thinking particularly of DNA origami, where the more components you add, in some sense, the more reliable it gets. So at the time that Paul invented DNA origami, myself and others in my group were struggling with systems that had just a few strands, 4 to 8, which was twice as many as we had done the previous year. But every little part of that system we had to look at, run gels, see whether it was working right, ask ourselves questions about each individual nucleotide. And when Paul went out and did origami, somehow there were overwhelming cooperative effects that swamped out the deviations that we didn't understand. And so there's some kinds of systems where sort of the more you build, the more cooperative, the more it behaves the way you want it to. And other kinds of systems, in particular small systems that are sometimes harder to engineer than large systems, where every part of it has to be right. So I think something worth thinking about is, beyond origami, what kinds of systems can we design where the larger it gets, the more robust it gets in a very natural way? So that's my thought.
Thank you, Eric.
I think my microphone works now. Can you hear me?
Yes.
Yeah. I think that's a very good point that Eric just made. When you're going into living cells, I just want to kind of make a quick point that living cells, you presumably they are going to replicate at some point. And given that we can't design living cells, replicating cells from the ground up, there are hidden parts in there where the overall fitness is defined by evolution and evolutionary dynamics. So this is kind of your nightmare as an engineer, Guy, where I'm giving you a system to engineer and I'm saying, well, there's part of it still mysterious, but don't worry about that. And so I think one of the things that we need to do is make sure that we can start to produce living cells that are able to boot up from the ground up. I don't think the synthetic biologists are so far away from that, but until you have that, you're still going to have a bit of mystery in the machine. And whilst you have a bit of mystery in the machine, you're not sure that some other evolutionary dynamic isn't going to be going on, and there are reactions and things you have not abstracted that will add to the cooperative effects that Eric has described. And I'm now talking about inside a living, replicating cell. If that makes sense, I don't know if that's a useful comment.
I think it chimes with what Richard Murray said yesterday, which was he thinks that there's more potential in completely synthetic things than in engineered cells. potentially partially for that reason.
Right. Anyone else wants to chime in? Before
Helga's had her hand up for a while.
Sorry, yeah, I can't see.
Hello. Could you hear me?
Yes.
Yep.
Yeah. So, cause my work, like, I'm kind of like a new PhD student. So my kind of work is looking to design a control and optimization system for gene expression of like my therapeutic use for my project. And when I look for to kind of design this kind of system, I realized there's a kind of, we have to balance the in vitro and in vivo use. So when we think about the in vivo or animal work or something, it's quite difficult when you think more like in realistic factors and how cells and other parts like interact with each other. Is quite stressed, like when you add more and your system or theoretical part, like your theories is more and more it's just like you can't really optimize that. That's kind of issues I have for now. And I don't know which way to go after that.
So are you saying that it's kind of difficult to move between, like, if you're prototyping stuff in vitro, it's kind of difficult to move into a much more complicated in vivo environment? Is that?
Yes, and because for those two, you even have different like different like requirements to say. So in vivo, you think about more like biodegradable bioavailability stuff. For example, in vitro, like in cells, you don't have to worry about too much about like cytotoxicity, immune genetics and stuff. So I think this part might be quite too far from what you're talking about for now, but I think for the gene expression control and optimization for a certain therapy, like potential therapy, is kind of something to think about.
Thank you.
Okay. Sorry.
So in the interest of time, I suggest that we move to the next question, and please do use the channel to add any comments or any questions that you wanted for this first question there. So the second question was, in what ways should we be looking to further emulate natural molecular systems? In what ways should we be trying to do things differently? I'm not going to go through the whole list, but we have captured a few of the points that were raised during the meeting, which I invite you to have a look at. And potentially, please do let us know if you have other points that you want to raise, or if you want to discuss some of the points that we've already put down.
Deborah, or if your point's still valid, crack on.
I'm not sure it's still valid, and I'm not sure how to write in whatever chat that I've been referred to. I'll just say my point because I don't think it's very long, but it makes me think that Yannick Rendelet's recent talk, right? Maybe we should be looking first for the kind of DNA nanotechnology that can go in cells and not have a function and just not muck things up. And maybe there's a nice way to just select naturally for certain DNA nanotechnology that are silent and compatible with the living state, and then mutate from there. And maybe if you could also remind me where to go to have text conversations so that I don't derail further talking.
Of course. So if I'm just going to show you directly. So on Teams, if we go to the workshop list of channels, there is one channel called Workshop Overarching Questions. And if you click on there, you have basically a channel where the questions are summarized, and you can basically type your points there.
If you just want to have a chat with somebody from the conference, you can use the chat button that's on the top left of the screen. If you just want to talk to one other person or to other people.
All right, sorry for derailing.
Anyone else about these points here? And I will just put it on the screen so that everyone can see them. It's a long list, so I'm not going to read it.
So I have a comment that's not as related to these points exactly, but is related to the top question. And also the previous question, and to some extent Eric's comments that, you know, larger systems might work better. And I'm immediately brought to mind about, you know, machine learning architectures where building a very small neural network can be hard, but once you get above a certain size, it works very well. And you wonder if real biology maybe has a flavor of that, where there's redundancy and multiple repeating species and motifs that do the same thing, and that makes it easier to evolve and more robust to perturbations. And maybe that's something we should try to emulate when we design systems, where if we put in natural redundancy and also sort of take an evolutionary or sort of heuristic methodology to design, instead of, say, designing every piece by hand, we say, let's have lots of pieces and sort of let them evolve to get where we want. Could we emulate sort of a machine learning type architecture to accomplish our goals? And would that naturally be more robust for the reasons Eric raised earlier?
Yeah, I mean, I've kind of written a new word down on the document now, which is more than redundancy we're talking about with machine learning, right? We're talking about emergence, which is you put in enough stuff together and you get qualitatively distinct behavior. Right, and this perhaps is touching on, again, what Richard Murray said. Sorry?
I don't think it's just enough. I think it's also has to be very particular. If you build random neural networks, they don't do very much very well. You have to have them evolved or trained or optimized towards a particular goal. So I think its size is not the only problem. We can build things that are big and random. We need to build things that are big and random and tune them in some way.
Okay.
I think there was a hand raised by Lee.
Yeah, I was just going to make a comment about emergence. It's a trigger warning. It's also a little bit I'm going to talk about in my talk. I think that if we can't, emergence in engineering systems is a bit of a kind of way of saying we don't know what's happening. So yeah, I'll talk about it in a moment, but I just was going to ask what you really mean by emergence, Thomas.
Well, yeah, to some extent I do mean
Incomplete
that.
information, just made up jiggling. I mean, emergence is kind of a word that people use when they don't understand stuff.
I do mean that, and I think you could make the case that the point of nucleic acid nanotechnology is it's rationally understandable. That's why people have been able to do some of the things they've been able to do with it. And that if you want to play the game where you make a big soup and then perturb the soup, why wouldn't you do that with proteins rather than nucleic acids? Maybe there is a reason.
No, I get that. I get where you're coming from, and that's really cool. But what I mean is there might be a way of more precisely understanding the starting conditions. Because certainly if you say, look, in a complex system, like whether it's a reaction diffusion system or birds flocking, there are structures which emerge, and that kind of hard emergence that you can engineer is, I think, what you're getting at, right? So I think it's like, are you saying you want to understand those control knobs a bit better to have that causal effect to get you that additional information processing, or something else?
Yeah, I think that is what we're saying. Like, have a way, like as Eric said, build systems that tend to behave like this, rather than just, right? It's not just build a big system. It's build a system that has this ability somehow baked into it. But yeah, it's pretty nebulous.
Andrew Tuberfield, there are many hands raised. I think I can see them only when people just raise them. After that, I can't see them anymore.
I think it's just Andrew and Tomislav.
Okay. I mean, training is rather akin to evolution. So what you're asking for is something that has the capacity to evolve to something that's useful. And the question then is timescale, or what's the right level of abstraction at which to try to create this thing. And do you create it with real nucleic acids, or do you create it using these wonderful simulators? So is the question how to interface the DSD to a neural network to evolve something you can later make? Or do you need 1 billion years to do it?
Okay. Tomislav?
Yeah, I'm kind of in between the two questions, but I would just say that I think it's very clear that living cells are basically spaghetti code. It is exceptionally complicated, or as a dynamical system, the ODs that describe the system are very complicated. And I think the only way to ensure that you can do something in the cell or control it is to have very, very detailed biological knowledge of exactly what is going on. I don't think that without that you can do much, except in very specific cases where we have well-defined and semi-isolated subsystem within the cell. And for number two, I would say that, so in mathematics, there is this famous problem called Hilbert-Sixin problem. And what it is that for those more mathematically inclined, you basically have two ODEs, only two, and each one has polynomial right-hand side with quadratic polynomial. And the question is how many limit cycles or how many oscillators this system has. So you have only two equations and you have simplest possible nonlinearity, quadratics. And this problem has not been solved for 150 years. And you can see that this is the kind of thing that cells and any synthetic system will have to do, whereby you will have a dynamical system and you will have to fine tune to have certain dynamical behaviors such as oscillations. So I think that general principles can only bring you so far, but ultimately I think that to reach the complexity and functionality of living systems, fine-tuning and a lot of trial and error is necessary. I think it's very difficult to do it systematically and in a simple way.
All right. So indeed, thank you very much, Tomislav. Please do think about this and do use the channel to input this. I will show you that you will all have access to this document as well. The last part is what key application features can DNA, RNA, nanotech in living cells enable? And that's where you can see that the list is a little bit shorter. So if you have more, please do let us know.
So I think there's the clear question of do you want the stuff actually in living cells, or do you want cell-like things that are doing something cool, but it's not something that used to be an E. coli. I don't know whether you want to distinguish between those two things. I don't think that's necessarily vital. I mean, Andrew presented a new application for sort of nucleic acid nanotechnology today, which was sort of combinatorial exploration of interesting chemistry, right? Is that a fair way to describe it, Andrew?
Yeah, absolutely. I mean, it's not that idea isn't new, but it's a new way of doing it.
Yeah. Eric's hands up again. Got in, Eric.
Yeah, sure. So just to go further in along your question of should we as a field be thinking about applications of DNA and RNA nanotechnology inside living cells versus designing systems that are maybe inspired by life but entirely non-biological and not working in living cells? I think that's a fine question, but there's another one which is maybe in between them, which is designing DNA nanotechnology systems that work around living cells and interact with living cells from the outside for applications. And you know, we've heard some of that already, but I think it's likely to be a very promising direction, simply because outside of living cells we have more control over the environment of our nanomachines, and there's plenty that one can do in the extracellular environment that is of importance to biology, whether it's detection or building scaffolds or what have you. So many people are thinking about that, but I think it's worth saying out in this context.
You wouldn't have seen this because it was one of the first talks on Monday, but Oscar says was very much talking about sort of bionic systems where you encapsulate a cell, and then and then outside that you kind of have an onion where you encapsulate some synthetic circuit, and the cell is providing useful things to the synthetic circuit and then getting stuff back. And he was talking about using chloroplasts as battery supplies and things like that. So that was very much in that direction. Will, your hand's gone back up, I think.
It has. I think that one of the things I don't see on here is sort of sustainability applications, and in particular, I think that when you want to produce products, especially at scale, from molecules, life has a very, it grows exponentially. There's a lot of good reasons why it might be useful, and I think it's very hard to give up the billions of years of protein evolution and the catalytic capabilities they come with. You still get to the question that Richard asked, could we sort of take those proteins and re-engineer them into something new? Possible, sounds very hard. I always think that one of the ways that life regulates itself the most is via chromatin structure. And this is one of those things that DNA nanotech actually could also do. And it's something I haven't seen synthetic biologists put fancy DNA nanotech to sort of modify the structure of plasmids or the genome of different organisms the way chromatin works in order to provide a robust regulation. So I think that there's ways that we can integrate DNA nanotech into cells that we haven't considered yet that would give us advanced regulations, but not make us have to sort of redesign all of biology.
Yeah. Yeah, very fair point. Okay, so this session, we could go on and discuss that quite at length. This session was really a session meant to seed ideas and to start the conversation going.